describing this whole process, oh, hang on. Not so you can go and be a cellular biologist, but so that you can understand there's a process that cells go through. That process can be faster or slower, but you see they have to copy an enormous amount of information, okay? Because of that, cells are going to make errors, all right? And that's going to become important as we go. Cells are going to have a mutation rate of their own, and we're going to learn things about, radiation, how radiation can influence or increase this already present mutation rate. If I asked you to go copy a book out of a library, I bet you could do it, okay? If I gave you plenty of time, you could probably copy that book with no errors, right? But if I told you to go copy that book today, right, you might make some errors. If I told you that copy that book within the hour, you're going to make even more errors, right? So the speed of replication matters. Good. So cells have to build proteins at some rate. Cells have to divide at some rate. They have a lifespan and they have to grow and divide. So there's a life cycle for a cell. And as we correctly said earlier, the average life cycle of a cell, about 24 hours. There's two broad categories here. We've got mitosis, cell division, active cell division. This is the point in time where the nucleus is going to change a little bit in its structure. It's going to make it so that the cell can copy all of its individual components, individual pieces of DNA, every other organelle for the cell, and get ready to divide. That's mitosis, active cell division. And if you look at the little, the pie chart here, mitosis is only a small chunk of the cell's lifespan. Okay, that active division, replication, and ready to divide process. The majority of the time is taken up by what's called interphase. Interphase, reproductive interim phases, are made of three different things. You have what's called gap 1, synthesis, and gap 2. Synthesis is of course protein synthesis, the cell living its happy life. And these gap phases are the cell just living its happy life with no synthesis. Again, the whole idea is that there is DNA, and we're talking about, we're going to need to learn how can the DNA be damaged. So things that are important here, structure of chromosomes. The structure of chromosomes changes depending on what stage of division that the cell is in. So during interphase, which is the synthesis and gap phases, no active complete cell division, the chromosomes appear only as a diffuse granular mass of loosely coiled chromatin, basically a bowl of spaghetti. Chromatin is just loosely bound DNA. So a series of bead-like globules of protein called histones around which DNA molecules are wrapped. To be specific, they wrap twice around each one of these little protein beads, and it's a way that the cell can package the protein, but loosely, but still in some version of an organized way. The entire strand is called a chromatin fiber. Strands of chromatin twisted together into a rope-like structure. That rope-like structure is what's going to eventually package up and be that X-shaped, double sausage-shaped chromosome. So we'll look at these three levels of structure here, okay? This is what's called a logarithmic scale, by the way, where they start out like zoomed really far in, and then they keep zooming out and out and out until you get down to, the thing that you could see through the microscope. So starting up top here, okay, you've got at the top left, you've got the most expanded view, right? the actual view of the DNA double helix, the twisted ladder structure. That twisted ladder structure gets wrapped around those protein beads called histone beads. We show them here as being like a perfect sphere. They're not a perfect sphere. You've seen what protein looks like. You saw the spike proteins. You saw hemoglobin. It's kind of just a glob. In fact, we use the word globule to talk about proteins. In fact, globin is a word meaning protein. Okay, so it's really just a glob, really a chunk of protein called histone beads. And the DNA strand actually wraps around those beads, okay, forming a long beaded structure called chromatin. That all packages up to be what's called a chromatin fiber. And if you follow it down and into that fiber, you'll see that there is a single side structure called a chromatid. Okay, so a chromatid is half of a chromosome, and it is basically just the packaged up, smushed up DNA structure that's going to connect in the center to the other side, to another side chromatid at a structure called the centromere. Centromere being the center where the X-shaped structure connects. DNA wraps around histone beads, forms chromatin strands. Chromatin strands twist together, forming chromatin fibers. Fibers loop into the shape of two chromatids, centered in the middle with a thing called the centromere. This doesn't always look like that. As we said, during the entirety of interphase, you can't see. So with a microscope and even the correct kind of dye to dye the cell structure, you can't see the chromosomes in the nucleus during interphase, which is the majority of the cell's life. You can't see those because they are loosely packaged like a bowl of spaghetti. Okay. Complete chromosomes appearing as a two-chromatid structure here before synthesis phase and duplicated here, okay, after synthesis phase. These only appear during a specific time in the cell's life. These most of the time will look like a loosely packaged bowl of spaghetti. I'm not going to talk about the whole cell division process. What I will hone in on here is the process called mitosis. Okay, mitosis, also called replication division. The idea with mitosis is you've got a complete copy of the cell structure, right? A single cell forms two identical daughter cells. And in order for that to happen, every component of the cell needs to be copied, including the entirety of the nucleus. We can divide the mitotic process, the cell division process, into 4 individual phases, subdivided 4 stages, called prophase, metaphase, anaphase, and telophase. I'm going to play you a video in a little bit here that's going to help with this. Prophase. The nucleus begins to swell. During metaphase, the nucleus elongates, chromosomes line up in the middle, and super interesting, fibers come in and attach to the centromeres of all of the chromosomes. They're called spindle fibers attached to the centromeres. When anaphase begins, step C, those fibers attached to those centromeres begin to pull the ends of the nucleus, pull on each end of those chromosomes. Those double chromosomes split and are pulled to opposite ends of the nucleus. During telophase, the new cell membrane divides the cell, splits, the nuclear membranes dissolve and reform in the new cell. And the new cell will go about building all of its new cell components. So, the video. This is going to help, I think.